Hey everyone, the brain. We're going to divide up this chapter 15, into brain and cranial nerves. Here in the brain, we'll be learning more about the nervous system, the cells that are found in it, but also what's protecting the outside of the brain, since it's such a vital organ. It has bone protecting it and other things. So let's take a closer look. Like I said, we'll be discussing not only the different brain structures, as well as the spaces within the brain, different fluids that help nourish and protect the brain, and then we'll also distinguish between different functional areas of the brain. We'll learn what the cerebral cortex is, and then subcortical regions. By the next part of the chapter, 15b, we'll look at cranial nerves. It'll be important to identify all these things on models as well as cadaver brains. So we'll have a quick look today in complete anatomy to look at the 3D model, as well as a little bit of Ackland's and other videos to look at cadaver brains. When we look at brain structure and function, it took us as human people a while to figure out what exactly the brain did. We knew it was important for overall intelligence and decision making, and just, you needed to live. But different parts of the brain are important for different things. One crude way of learning of what the brain does is to learn through accidents and damage to the brain. For example, there is a man named Phineas Gage. Maybe you've heard of him if you've taken a psychology course before. And he was a railroad worker. I forget the year, probably 1800s. And when you work with railroads, you work with explosives so that you can put things in the ground. I'm not a railroad worker expert person, but they work with explosives and metal rods. And there was an accident where this metal rod exploded, but with the explosives, it catapulted this rod through his skull. And this is a 3D model rendering of that rod going through his skull. Now, if you saw a person with a rod through their skull like this, you'd probably think they were a goner. But he survived. He lived after this accident. So that tells us at least one thing here. This region of the brain that was damaged, while it might be important for something, it is important for something, it's not important for survival. For the basic core things you need to live, it is not important for that, because overall his physiology was fine. There were noticeable changes besides the obvious scarring in his face. There were some noticeable changes in his personality. He was very unpleasant to be around, and he was pleasant before. He was very rude. He said whatever he wanted. He was a giant jerk, to put it lightly. What this tells us is that this part of the brain then, while it might not be important for the most vital things like regulating your heart rate or telling you when you're hungry, it can regulate things that we consider personality, things like decision making, things like inhibition control. So we're going to go through different parts of the brain, and I'll point out where I can, where we've learned through studies of damage or loss of how we've come to know what different brain regions do. We all have heard of the brain. We've all seen pictures of brains, but what is it really? It is the center of our nervous system. Our nervous system has a central nervous system, and that's the brain and spinal cord, but even the spinal cord will relay information to the brain. So this is the true center of regulating everything in our nervous system. And it regulates pretty much every organ in your body. Your organs can have a separate type of regulation. You'll learn about autocrine, paracrine signaling when you get to physiology, but the nervous system can regulate a lot, most if not all of your body in some way. It's far more complex than the spinal cord, 20 billion neurons. There's a lot of neurons communicating with each other. One reason why you need multipolar neurons in the central nervous system. And since it's part of the nervous system, since it uses electrical signaling combined with chemical signaling across synapses, you have rapid coordinated responses to changes in the environment. Because it's such a vital organ, just like your heart and lungs, you need protection. Your heart and lungs have your sternum and ribs to protect. Your brain has cranial bones. We've gone over those cranial bones: frontal, parietal, occipital, temporal, sphenoid, ethmoid. All those bones are protecting your brain directly. In addition to that, your brain needs another layer or layers of protection. Your heart and lungs, because they're moving all the time, they have serous membrane. Your brain isn't moving all the time, but you need this you need layers of protection that are unique for protecting the brain and spinal cord. We're going to talk about the meninges and the fluid inside of it that helps protect and nourish your brain and spinal cord. So to start with, what is actually protecting the brain, besides the bone, are something called meninges. If we take a look at the scalp here, we're looking from a superior view, you can see here's the skin. Skin means epidermis, dermis, and hypodermis. So you remove that. Next layer is that epicranial aponeurosis, that special muscle-to-muscle tendon that's connecting the frontalis muscle to the occipitalis muscles. Aponeurosis is dense, regular connective tissue. Remove that, you get periosteum. You get periosteum covering the bone. And then you get to the bone itself of the cranium, whether it's frontal, parietal, temporal, occipital. Deep to that, the next layer down, you get to something called the dura mater. Next layer below that, you get to the arachnoid mater. Next layer, you get the pia mater. There are three layers, these three here: dura mater, arachnoid mater, pia mater. These are part of what are called your meninges. Meninges is a funny word. I believe it's Greek. Meninges is plural. In case you see this, the singular for meninges is meninges. It follows the same pattern as phalanges. Singular phalanges is phalanx, ANX. Singular meninges is meninges. Anyways, these are the three layers that are covering your brain. The pia mater directly covers the brain. It's the deepest. It's super thin and delicate. You can't really peel it off of your brain. It's just, if you ever see a bare, quote unquote, bare brain, you're looking at a thin layer of pia mater covering the brain. Pia means soft. Pia means soft. Arachnoid, maybe you've heard of that word before, like arachnid. Arachnoid means arachnid-like. Arachnid is a spider. It's kind of like a spider web. It's web-like material. Mater means matter. Web-like material. It's also thin, but more like, the way I kind of picture it, it's kind of like plastic wrap over the brain. It holds down blood vessels. It's thin. You can kind of see through it. It's a little hazy. With enough force, you can tear it. So it's not as delicate as pia mater, but you can peel it off, and it's flimsy and it's thin. You can kind of see through it. We'll take a look at this later. You can see, for example, if you take a look here at the arachnoid mater, you can see blood vessels hiding underneath the arachnoid mater. It's visible through it. The dura mater, dura like durable, it's very tough. It's the thickest, toughest layer, most superficial layer of the meninges, and it's protective. So you have dura mater, arachnoid mater, pia mater. Let's take a closer look at what each of them do. So the dura mater, like I said, is the thickest, most superficial. It has two layers. The The first layer, the periosteal layer, coloring it down here in this inset. The periosteal layer is just continuous with the periosteum. It is the periosteum of the cranial bones, frontal, parietal, temporal, occipital, and so on. It's fused to the periosteum of the cranial bones. If you touch an infant's fontanelle, you can feel this layer. And so we've learned what periosteum is made of. Periosteum is made of dense, regular connective tissue. So it's there to help provide a connection between the meninges and the bone that's the harder bone that's protecting it. The deeper layer, the meningeal layer, which in this inset is right here. The meningeal layer is the innermost layer, and sometimes it diverges from the periosteal layer. You can see that in some places it's together with the periosteal layer, but then sometimes it spreads apart. In the places where it spreads apart, it forms a space for blood vessels. Where I'm coloring right now, that's a space for large veins, large blood vessels. These large veins drain blood away from the head. Collectively, they're known as dural because they're within the dura mater. Dural venous, venous is the adjective for a vein. Sinuses. The word sinus, we've talked about with paranasal sinuses. The word sinus just means large space. So in this case, a large blood vessel. Dural venous sinuses can be found within those spaces where you get a separation between the periosteal dura mater and the meningeal dura mater. So overall, the dura mater is protective and can provide spaces for blood vessels to drain blood away from the head. Your brain is divided into two hemispheres. Your brain is divided into two hemispheres. In this picture, and also in the picture below it, we're showing a mid-sagittal cut. And so the brain would normally fit in, let me use a different color. The brain would normally fit in right here. And there would be another half on the other side. The two halves are connected, and where I'm coloring right now in green, there is a connection between the sides of the brain, but there's a bit of a split. So it's kind of like a half split. Like if you took an axe and chopped a piece of wood, but it didn't split all the way, you'd have the axe in the middle a little bit. That's what we have right here. What I'm coloring in here in green is where dura mater splits in between the brain, but not all the way. It splits into that mid-sagittal area. The name of the mid-sagittal area between the hemispheres of the brain, the name of that is the longitudinal fissure. Longitudinal because it's long, fissure because it's a deep space. It's a space between the left and right hemispheres of the brain. We're going to learn the name of that. That's called the cerebral cortex. The term for the dura mater that's in the longitudinal fissure, the term for the dura mater that's in the longitudinal fissure, it's called the falx cerebri. Falx means crescent, so it's kind of crescent shaped. Cerebri is referring to the cerebrum. The cerebrum is a large, wrinkly part of your brain. There are more parts to your brain than the large, wrinkly part. It's the most obvious part of your brain. So when you think of brain, that's probably what you think of, but there are other parts that we'll go over. So the falx cerebri provides support in between the hemispheres. There is a major dural venous sinus running through it as well. And it needs some way to attach on anterior and posterior sides. We have learned the attachment on the anterior side. On the anterior side, the crista galli is an attachment point. Remember that little crest of the ethmoid bone? The falx cerebri attaches to the crista galli of the ethmoid bone on the anterior, on its anterior portion. All the falx cerebri is, if I didn't make this clear already, the falx cerebri is fused dura mater. So you have dura mater on either side. The dura mater comes together here in the middle. The fused dura mater that goes in the longitudinal fissure, that is the falx cerebri. And that's what you can see down here. Falx cerebri. We have our periosteal layer. Of the dura mater. Here in this picture, we have the meningeal layer here. And then you can see one example of a dural venous sinus. Don't worry about the specific name of it. There's multiple specific ones, but we'll collectively call them dural venous sinuses in between the two layers. The next layer down is the arachnoid mater, which I'm going to color in yellow. Now it has what I'm coloring here in yellow, but it also has the web-like portion of it, has these extensions that reach down deeper. The arachnoid mater, because it has those web-like things reaching down, it creates a space. Let me highlight that space in red. Let's try red. It provides a space. That area is called the subarachnoid space. It's below the arachnoid membrane. It's a space. That subarachnoid space is space for fluid. Fluid, water, is super cushiony, helps distribute important nutrients or gets rid of waste, just like blood. Blood is made of mostly fluid. So here we have the space for fluid, and the fluid that runs through here, it's called cerebrospinal fluid. It's called this because it's shared between the brain, the major part of the brain, the cerebrum, and your spinal cord, hence cerebrospinal fluid. Because of those fibers made of collagen, collagenous, am I saying that right? Collagenous fibers are anchoring down to the pia mater. You're also getting blood vessels to stay pinned down underneath. I pointed out earlier that you could see blood vessels hiding underneath the arachnoid. Blood vessels are hiding underneath the arachnoid. We'll talk about this cerebrospinal fluid more once we talk about ventricles coming up, but we need space for this special fluid to flow around, and here is that space. The deepest layer let me recolor in arachnoid. That here in yellow is arachnoid. And then in green, I'll do both layers of the Dura mater in green. The deepest layer, thinnest layer connected to the surface of the brain, and it goes along with every crevice. This is the pia mater. If you notice, the arachnoid mater did not go with every crevice. It just kind of covers, like plastic wrap, it just kind of covers all the crevices without actually going in every little nook and cranny. The pia mater does go with every wrinkle and fold. The term for a groove into the brain, the term for the deeper part of the wrinkle, plural is sulci, singular is sulcus. So a single groove is a sulcus, plural sulci. When it comes up, so sorry, here's a sulcus right there. When it comes up, the peak, that peak is plural gyri, singular gyrus. So the outer part of the brain, the cerebrum is made of gyri and sulci, and the pia mater follows every single gyrus and sulcus. The way that pia mater attaches to the brain is it's anchored by astrocytes. So there's one function of those special glial cells that we find in the central nervous system. They help anchor larger blood vessels of the cerebellum. Let me point to one. You see blood vessels here anchoring down, also with the help of the arachnoid. Together, the pia mater with these astrocytes, why they cover every single little wrinkle of the brain, or what they're doing, they're helping create a blood-brain barrier. Now, I'm sure you've heard of this term before, but what does this actually mean? Our blood for our whole body is, of course, super important. We need to get oxygen, we need to get nutrients, we need to get rid of wastes, among other things. The brain uses a ton of nutrients, uses tons of oxygen, needs to get rid of a lot of waste. So we need constant circulation to and from our brain. But the thing is, our blood, in general, is way more susceptible. It is fairly susceptible to be exposed to things. Like, let's say we eat a meal that has been spoiled. That spoiled toxin from bacteria or something else might get into our bloodstream and could hypothetically damage our brain. Now, we don't want that. We want another layer of protection. There's already a layer of protection with our liver to help process stuff in our blood, but we want to be extra cautious. Our brain has an additional, you could call it a layer of filtration before we get stuff in the blood into our brain. We're going to see that extra layer of filtration, how we take the important things out of blood and form cerebrospinal fluid. You can think of cerebrospinal fluid as a super highly filtered interstitial fluid taken out from blood. But what we don't want is to just take anything that's in our bloodstream, viruses, bacteria included, and have it exposed to our brain. We want to minimize that possibility as much as possible. So we create this barrier. To create this barrier, most blood vessels, there are some exceptions, and we'll go over those exceptions later, but most blood vessels that are supplying the brain are actually completely covered by astrocytes. If you take a look down here in these pictures, we look at an inset of a blood vessel. These astrocytes have these extensions, and they're covering every part of a blood vessel, and there are tight junctions to prevent anything from leaking out, because blood vessels are leaky. So we're keeping everything in the blood vessels, and we'll have the opportunity at the right time to take stuff out from blood where we can filter it better, where we can take all the good stuff but leave the bad stuff. So tight junctions help regulate and regulate what's exiting from the blood, pretty much preventing anything from exiting blood, and so that we form this barrier, this blood-brain barrier. So where can stuff from blood be brought in? Where can all the good stuff, nutrients and other things be brought in from blood? Take a look at this blood vessel here. If we have blood coming in, there's certain regions within our brain where we can take nutrients out. We can take out ions, vitamins, nutrients, oxygen. The cells that take the good stuff, remember ependymal cells? Ependymal cells do this. What I'm highlighting here are a bunch of ependymal cells. Ependymal cells take stuff out of the blood and bring it out into this specialized interstitial fluid known as your cerebrospinal fluid. Cerebrospinal fluid is formed, therefore, when ependymal cells take nutrients and fluids out from the blood and bring it into this space within the brain. There are spaces within the brain. The term for, it's not every ependymal cell that does this. There are certain regions within the brain where ependymal cells do this. The term for the ependymal cells that do this within the brain, this is collectively known as the choroid plexus. Choroid, you're going to see this word come up a lot. Choroid refers to a region with blood vessels. Plexus means a network. So it's a network of blood vessels with cells, ependymal cells covering it. That's really what it means. When we see it we can see this in cadaver brains. It just looks like this dark ball of blood vessels with stuff covering it. And that's exactly what it is. When nutrients are taken out, let me trace this over with green. When nutrients and fluids are taken out and brought into the brain, they're brought into a space called a ventricle. They're brought into a space within the brain called a ventricle. We'll see this term ventricle also in the heart. It just means a big space. What happens is, take a look at this picture at the left. You have choroid plexus in several parts in your brain within ventricles, which I've colored in pink there. There's choroid plexus here, there's choroid plexus here, in all of your ventricles. Cerebrospinal fluid is produced from these ventricles, and all the ventricles are connected. These ventricles connect to a central canal within the spinal cord and can travel down the spinal cord as well. It's all connected. That central canal eventually leads out of the spinal cord. Let's go this way for fun. And now we're in the subarachnoid space. Ventricles of the brain, within the brain, and the central canal within the spinal cord, that space is connected with the subarachnoid space. So all of this space is just filled with cerebrospinal fluid. We're constantly producing cerebrospinal fluid, so it's going to flow in one direction, and we're going to see it exit in another direction. It's going to exit in special, we'll call them drainage areas. Exits in special drainage areas called arachnoid villi. You might see them called arachnoid granulations. We're going to call them villi. The word villus singular is villus. Villus means finger projection. So arachnoid villi are finger-like projections of the arachnoid mater. Look at this top right illustration. Here is the subarachnoid space, this space right here. Cerebrospinal fluid will come to the subarachnoid space and then empty out this little protrusion. This protrusion that I'm going to color in green, that is the arachnoid villus. And the arachnoid villus pokes past the arachnoid mater into the dural venous sinuses. So this space that I'm going to color in orange, this is a dural venous sinus. This is a dural venous sinus. You have arachnoid villi poking into dural venous sinuses. And any fluid from the subarachnoid space, any cerebrospinal fluid can drain out into the bloodstream. We've formed cerebrospinal fluid thanks to the choroid plexus. So fluid was taken out from the blood, and now here fluid is exiting back to the blood, out these arachnoid villi into the dural venous sinuses. In that way, we're getting constant flow. We're adding fluid, removing fluid. Adding fluid rich with nutrients, removing fluid that has waste. This is a circulatory system of the central nervous system. The 4 ventricles where you can find choroid plexus, they have names and they're found in very specific places. The first ventricle is called, well, the first 2, I should say, these are called lateral ventricles. There are 2 of them. There's one on your left side, which I've highlighted here, and then there's one on the other side, highlighted here. Those are your lateral ventricles. We'll get a better view in a minute when we look at complete anatomy. They arch from the anterior side to the posterior side and then back anterior again. There's a thin separation between the 2 hemispheres. So the 2 hemispheres are connected. If we see, if I can draw from a frontal cut, if this is one lateral ventricle and here's another, they are connected. Like this. We'll call this the right lateral ventricle, the left lateral, if we're looking from a posterior view. They are connected this way, but there is a separation between the 2 right here. That separation is known as the septum pellucidum. Septum means wall. Pellucidum, lucid. Lucid means clear. It's a thin, clear wall. Clearish. Thin, clearish wall. The lateral ventricles then lead to the 3rd ventricle. The left and right lateral ventricles are 1 and 2, no particular order. They're just known as the lateral ventricles. So the next one is the 3rd because it's the 3rd one in line. The 3rd one is located deep in the brain in this region called the diencephalon. We'll learn what that area means exactly, but in our picture down here, it's this circular-ish region. It has a funny little hole in it. And then from the 3rd ventricle, we continue down the brain, and we reach the 4th ventricle, the last one. And the 4th ventricle is kind of by that little brain in the back, the cerebellum, it's down here. It's a bit more narrow. Four ventricles, left lateral, right lateral, third, fourth, and they're all connected. Each ventricle has choroid plexus, so each ventricle can produce cerebrospinal fluid. Putting all that together, here's that frontal coronal section that I was drawing earlier. Putting all that together, we can see complete flow of cerebrospinal fluid through our brain and spinal cord, through our central nervous system. If we draw choroid plexus, it's found in all ventricles, but let's say, just for the sake of simplicity, let's draw some choroid plexus in the lateral ventricles. I'm drawing it in the picture on the left and also drawing a little bit in the picture on the right. Drawing some choroid plexus. So from there, we can get cerebrospinal fluid produced in the lateral ventricles. From the lateral ventricles, we'll pass through a very narrow passage. They're called interventricular foramina. Interventricular because they're between ventricles. Foramina, plural of foramen, holes. These little passageways, those are interventricular foramina. Then we get to the third ventricle. There's only one third ventricle, and it's very central to the brain. Then we go to a very narrow passageway. That super narrow passageway is known as the cerebral aqueduct. It's at the level of the midbrain. We'll learn where that is. Then we get to the last ventricle, the fourth ventricle. Down here. And then from the fourth ventricle, we get to the central canal. Central canal goes all the way down to the end of the spinal cord, and when we exit the spinal cord, we get to the subarachnoid space. So that's going to be out here. From the subarachnoid space, we can go through arachnoid villi out to the dural venous sinuses. And so that's how we get complete flow of blood through the brain. Blood gets filtered, so to speak, by the choroid plexus, by epithelial cells of the choroid plexus, and we get flow through the brain. And then we exit that fluid which is now accumulated waste, out the dural venous sinuses. Just as there is circulation of blood through our circulatory system throughout our body, there's circulation of cerebrospinal fluid through the brain and spinal cord. It's not going to be as high pressured since there's no heart pushing this fluid through, but there is movement of fluid throughout to circulate nutrients and get rid of wastes. To supply blood to the brain in the first place, or to remove blood away from the brain, let's go over the specific blood vessels that deliver blood to and from the brain. The major arteries, the artery, an artery is a blood vessel that delivers blood from the heart out. So in this case, from the heart to the brain. The two major groups of arteries are the internal carotid arteries and the vertebral arteries. Internal carotid are on the anterior side. They come through the anterior cervical region. Vertebral, as the name suggests, come by the vertebrae. They're on the posterior side. There are two sets of each, two internal carotid, two vertebral arteries. Vertebral arteries, you actually know what they are protected by, what bone marking they pass through here in the back. They pass through the transverse foramina of those cervical vertebrae and then enter into the brain through the foramen magnum. We'll take a look at that more in the class when we see the skull. So that's what delivers fresh oxygenated blood and nutrient-rich blood to the brain. Internal carotid arteries, vertebral arteries. From those dural venous sinuses, now we're getting blood away from the brain. From those dural venous sinuses, there's several of them. There's one here in the middle, there's several on the side. They drain to internal jugular veins. Jugular veins are what drain blood away from the body. They're on the anterior cervical region. Or sorry, away from the brain. They're in the anterior cervical region. I mention these blood vessels, these specific names of blood vessels, because they're super important for supplying blood to the brain and getting blood away from the brain. And if you were to clog one of them, if a clot were to occur, some kind of blockage of blood flow, that would be very serious. You need constant oxygen, constant blood flow to your brain, constant removal of waste away from your brain. If you get a blockage of blood flow, that can lead to a stroke. The long technical name of stroke is a cerebrovascular accident. Cerebro referring to the brain, vascular referring to blood vessels, accident because it's not something that you want to happen. Cerebrovascular accident. If you have a blockage of blood flow for one of these arteries, that will lead to death, to cell death, and potentially overall death. Strokes can cause damage to different areas of the brain. Another way that we are able to tell which parts of the brain are important for which things. For example, there are people who experience strokes who lose their memory. There are people who experience strokes who lose their ability to speak. And there are specific areas of the brain related to those types of things. Let's take a look at complete anatomy, a 3D model of all of this, of the different ventricles and areas where cerebrospinal fluid flows. And then we can take a look at a cadaver for the different meninges. If you go to our Canvas page, videos of brain and cranial nerves, scroll down to look at the complete anatomy screens. There's a lot of useful screens here. The one screen that I'm going to click on right now is the meninges and ventricles screen. So here we have our screen. You can see the brain and spinal cord have been isolated, but what we've isolated here is, we can see covering the brain and spinal cord is the meninges. I've cut the brain in half so that we can see other structures inside, but what's surrounding the entire brain and spinal cord superficially is the dura mater. So here's dura mater that's covering part of the brain. Here's dura mater that's covering part of the spinal cord. The dura mater of the brain and spinal cord is all one continuous piece. They've separated it here because you can name them different things, but it's all one piece. That fused dura mater in the longitudinal fissure, that's the falx cerebri. That's what we see here. You can see that it forms a crescent shape. So it doesn't fully separate the hemispheres, but it is between the hemispheres within that longitudinal fissure. You can see an example of a dural venous sinus sitting in a little gap between the dura mater. If we were to hide, let's hide a layer of the dura mater. So here is a layer. I'm going to hide this. The next layer down is the arachnoid mater. This would also be true. Whoops, that's still dura. Oh, that's we took off the periosteal layer. Now here's a meningeal layer. We hide that. Great. And now we have arachnoid mater. Arachnoid mater is a thin film covering the brain. You can see blood vessels underneath it. There are little arachnoid villi that we can actually point out in a better model besides this, that will drain into the durovenous sinuses. If we remove this, we can get to the pia mater. If we remove the arachnoid mater, sorry, it's slow. We remove the arachnoid mater, we get our final layer, the pia mater. It just says frontal lobe here. We can assume that the pia mater is directly covering every gyrus and sulcus. So it doesn't say pia mater here, but we can assume that it's directly covering it. We can't peel off the dura. We can't peel off the pia mater like we can the dura and the arachnoid mater. I showed you this for the brain. This is also true for the spinal cord because it's a one continuous piece. If I peel away the dura mater of the spinal cord, underneath it, deep to it, we would find arachnoid mater. No, it's not actually shown here, but there would be arachnoid mater, and I can show you that in another video. What we can also show here are the ventricles. You can see one of the lateral ventricles. From this lateral view, this is the left lateral ventricle. Let me highlight the whole thing. That's the left lateral ventricle, and there's one on the other side. If I clear this and move around, you can see the other one right here. That's the right lateral ventricle. The two ventricles are connected. You can see the interventricular foramen here on the left side. There are two interventricular foramina, one on the left, one on the right, and they connect to the third ventricle. The third ventricle is this circular-ish region. It's got a hole in the middle. We'll get to why later. There's the third ventricle. Here's an anterior view, lateral view, posterior-ish view. The third ventricle leads down through a narrow passage called the cerebral aqueduct. They call it mesencephalic aqueduct. We'll call it cerebral aqueduct. And from the cerebral aqueduct, we go down to the fourth ventricle. From the fourth ventricle, we'll get to the central canal. I need to hide part of the spinal cord so that you can see it. Let's hide this. And through the spinal cord, you can see this really long tube. I'm trying to select. I'll just point it out. But you can see this really long tube. That's the central canal. And the central canal runs all the way up and down the spinal cord. That's where the cerebrospinal fluid will eventually exit out into the subarachnoid space. You can see it continuing here in the center of the spinal cord. I know we've discussed the central canal for bone. This is central canal of the spinal cord. Different things. Switching over to some cadaver videos. We have videos in Acklins that we can view. So I encourage you to look at those. But I'm actually going to switch to a video from University of Utah, I believe. Yes, University of Utah. It's in this neuroanatomy playlist. The one regarding, it's the fourth one in the playlist, Meninges Neuroanatomy Video Lab. What you can see here in this video, these are two whole brains. One we see from an anterior view, one we see from a lateral view. In this lateral view brain, we can see a large piece of the dura mater is covering the brain. It's a very stiff, tough, that's what I want to say, stiff and tough material. If you remove the dura mater, you'll find this type of flimsy, thin membrane. That's arachnoid. It would, of course, cover the whole thing, but they've cut a piece of it so that you can see the different layers. And then deep to that, directly covering every gyrus and sulcus, that's pia mater. You can see, here's a close-up. Dura mater has been folded back. Arachnoid mater is covering the gyri and sulci, and you can see blood vessels deep to it. Underneath all of this is where you'd find cerebrospinal fluid. It's the subarachnoid space where you'd find cerebrospinal fluid. And we can actually see that in this one Ackland's video. Let me share it. What they've done, we're zooming into a brain, and there is arachnoid on top of this brain. They're going to inject just deep to the arachnoid mater, and you can see this subarachnoid space fill up with fluid. The subarachnoid space is filling up with fluid. That's where the cerebrospinal fluid would reside, and how it would drain out to the dural venous sinuses. There's plenty more to show. So I encourage you to watch the videos in the Canvas page through Ackland's, through University of Utah and British Columbia. But in the meantime, we'll stop here and look at talk more about the brain in the next video, more about brain regions. Thanks, everyone. Let me know if you have more questions. Leave questions in the comments, in the discussion. I'll talk to you all later.